Hey guys, hi. So this is the third video for the IBO 2013 PYQs. So this question is fairly easy. So apoptosis versus necrosis. So apoptosis is programmed cell death. That means this is a very planned activity and cells do it when they have either experienced extensive damage or damage from DNA replication. Maybe their telomeres have shortened a lot, or there's a pathogenic infections. Cells go to die themselves. It's cell suicide, but in a very planned way, and nothing spills outside. Okay, whereas necrosis is a trauma. So when necrosis happens, the membrane breaks, everything is spilled outside, which is later on cleared by the phagocytic cells. Okay, so apoptosis is induced in immature T cells that recognize self antigens. Now you must be aware about how we have several lakh types of antigen receptors in T cells. So by alternative splicing, which is cutting the mRNA segments in different ways, what we try to do is have several combinations of RNA, and this gives us the variety of the antigens that we have and the antigen receptors that we have simply. Okay, but the variety can include such antigens that can detect or that can combine with self antigens. That means proteins that are there on our own cells. We don't want such T cells for which have the receptors for self antigens, because that means it will start attacking our own cells. Further, this is what leads to autoimmunity if we are not able to control this thing, right? So such cells which have the receptors for self antigens, they are marked for death. Okay, that is where apoptosis happens. One of the cases where apoptosis happens. Intestinal epithelial cells losing contact with basal lamina undergo apoptosis. Now, how does our cells ensure that they don't turn cancerous? So there is something that is called as contact inhibition, and a need to be adhered to the surface. So there is a certain degree after which the cells, if they are coming in contact a lot with each other, they will be marked for death. Now where does this happen? This happens when a tumor forms, they lose the surface and they huddle together. We don't want that happening. So this is one of the cases where the cells are marked for death, and the cells must be adhered to the basal lamina, the part of the basement membrane. Now if they are losing the contact, those cells are once again marked for apoptosis. The reason is, if they have lost contact, they are metastasizing. Metastasizing means these cells are entering the bloodstream and will invade other tissues, which we do not want to happen. Okay, neural stem cells undergoing apoptosis expose on their surface a signal promoting phagocytosis. Now, as we just talked about, apoptosis is a planned activity. What happens during apoptosis is the DNA condenses to form chromosomes. After this condensation, small fragments are generated. Small membranous fragments of the cells are generated. These cells are engulfed by phagocytic cells like macrophages. Okay, so yes, if the stem cells or any cells are going to undergo apoptosis, they will undergo this process. Okay, so apoptosis is induced. Oh, we didn't mark it, but yes, that's true. That's true. That's true. Necrosis often induces an inflammatory immune response. Of course, they do. Now, why does that happen? Because you can imagine, when a cell bursts, it is going to release everything that's inside. It is going to release ATP. It's going to release DNA, and several other factors. This flood of chemicals often activates the immune responses and causes inflammation, swelling. Okay, so that's also true. So all true. This was fairly an easy question. See you next time with another problem to solve.